so we're gonna. Did you start it? Okay. I'm basically gonna give you a bunch of terms. You've heard me say most of these, but I just want to make sure that we know what they are. So the first one is allele. That's basically the trait that we're talking about when we're talking about genetics. When you do a Punnett square, so if, like yesterday, we were looking at the hairless guinea pigs, that having hair or no hair would be the trait. So the alleles would be these, the letters that we have here, the big H and the little h. Big H means that it is what?
Dominant.
Dominant. So traits can be, or alleles can be dominant, or they can be recessive. Recessive only shows if what?
Few plus recessive.
Yeah, you have two recessive. You have two recessive alleles, then it will show. Any other time, the recessive stuff is not going to show, because if there's a dominant, it always overpowers the recessive. So whenever you have gametes that are forming, do you remember what a gamete was? That's your sex cell.
Yep.
So for a female it's egg, and for a male it's sperm. So whenever you have gametes that are forming, they're only going to carry one allele for that trait. Now you have to have two for the trait. Why would you only have one in a sex cell? Because you get one from the mom and one from the dad. So gametes are your sex cells. Remember, they have half of your genetic information. So they're only going to have one allele for whatever trait we're looking at. Then, so if that was the mom's, then the dads would have one allele. When it becomes fertilized, what does it become?
A zygote.
Zygote. So a zygote is just a fertilized egg. And then it's going to have two alleles, because you had one from the mom and one from the dad. Okay, so I went over these terms with you yesterday, but just to make sure that you remember them, you have homozygous when we're talking about traits or alleles. Homozygous can be dominant, or it could be recessive. Okay, if it's homozygous dominant, it would be like HH, big H, big H. Recessive would be two lowercase h's. I'm just using h's because they're easy. You can use any letter that you want to. You don't have to use a certain letter. So if you ever have to make up your own Punnett square, you can use whatever letters you want. It doesn't matter. I would suggest using something though that has a very distinct big H or big letter and little letter, capital and lowercase. Because if you start using like S's, like if you cross S's, you know, it's kind of hard to tell when you start doing it all if that's a big or a little. So make sure that you can distinguish what it is. Okay, and then we have heterozygous. That's a big H, little h. That is still heterozygous dominant because it has a dominant trait in it. You can't have a heterozygous recessive because it's got a dominant trait in it, so it has to be dominant. So heterozygous is always dominant. Homozygous can be dominant or recessive. Okay, a couple of laws. So we've got the law of segregation. So this is talking about when gametes are forming. So you have two traits in you. So if we're talking about like hitchhiker's thumb. Okay, so if we're talking about that, you have a trait from your mom and your dad for how your thumb looks. So if it is straight or if it bends way back. Okay? When your body starts forming sex cells, those two alleles for the trait are going to separate. And then you're going to pass one along in the gamete that forms. So in the sex cell that forms. It doesn't always split the same way. So one time you could pass along the trait for hitchhiker's thumb, and next time you could pass along the trait for no hitchhiker's thumb. That's called the law of independent assortment. So the law of independent assortment says that alleles separate. So this will be during the formation of sex cells. So this will be during what? Mitosis or meiosis? Meiosis. So the alleles separate independently when forming gametes. And it's not always the same every time. So you're not going to pass the same traits along every time. You could. You don't know how it's going to separate out. Technically, you could pass the same trait along every single time. It's a 50-50 chance. Or you could pass it along every other time, whatever. There's no way to tell which one you're going to pass along. Last thing, and we kind of talked about these yesterday, just want to make sure that you understand it. So we are going to be using a lot with Punnett squares. So the basic Punnett square is just showing you what traits that are being passed along genetically, and you can tell from a Punnett square what is going to show up in the person. So that would be, you can see the genotype. Genotype are just the genes that are present. And then you've got phenotype. What is that?
Yes.
Yeah, that's what shows what you see. Okay, and remember, if it has a dominant in it, the dominant always supersedes the recessive. So if we do this cross, you're just listing what is meeting in these squares. You always put the big letter first. So if we did this cross with two heterozygous parents, so they have a dominant trait, but they're both carrying a recessive, you're going to get a probability. Does everybody have this part? Genotype and phenotype?
Yeah.
Okay. And we went over this yesterday, so this should be a review. So you're going to get a probability. And this is for every time a baby is made. Okay? And I keep harping on that because a lot of times people will say, if you have 4 kids, one's going to be homozygous dominant, 2 are going to be heterozygous dominant, and one's going to be homozygous recessive. That's not the case. This is the probability every time you have a baby. you're going to have a 75% chance, if we say this is hitchhiker's thumb, a 75% chance of getting hitchhiker's thumb, or a, what percentage for this one? 25% chance that they will not have it. Okay? So, probability is just the percentage for each time that a baby's made what it could be. So, each block in this is 25%. So, you just look at it. If it's asking for the genotype for this, we're going to list what the genotypes are, what the probability of genotypes are. So, if we say homozygous dominant, what's the percentage that you'll have a homozygous dominant genotype? Homozygous is
50.
same dominant, so two big letters.
50.
Two big letters.
Oh, yeah.
So that would be 25%. Heterozygous dominant would be what?
50.
50. And then homozygous recessive would be what?
25.
25%. So that would be the genotype. Phenotype, and what did I say we were doing? What trait did I tell? Okay, so if we're doing hitchhiker's thumb, what percentage would have hitchhiker's thumb?
75.
Okay, so 75% would have it, and then 25% wouldn't. And that's just an example. I can't remember if hitchhiker's thumb is dominant or recessive. So,
I think it's recessive.
so yeah, that might have been a bad
Because
example.
I don't have it.
You don't have it? Does your mom have it?
No.
You could have done
Wait, do your parents have it? Do you know?
Neither my dad does.
I don't think either of them had it, but you should have done Widow's Peak.
Okay, well, let's change it to Widow's Peak. Are
Or
you
how
saying,
about we
is
just
it
change
dominant?
it to like little bit pinky or something?
A bent pinky. Okay, yeah, Lachlan's got a bent pinky. So if we were doing bent pinkies on this, we'd have a 75% chance that the child would have a bent pinky and a 25% chance that it wouldn't. Okay, so this would be the phenotype. That's what you're seeing, what's showing up. Genotype is what you're seeing in the genes. You understand the difference?